AP Biology video. In this video, we are going to jump into Unit 6, which is all about gene expression and regulation. And the first topic that we're going to cover in this new unit is going to be kind of a throwback to our very first unit when we talked about our biological molecules. Today, we are going to review the basics of DNA and RNA structure and look at how exactly we encode all of that information to make our proteins and all of our structures within that genome. So, just to start back with a little bit of a similar slide to one that I included in that Unit 1 presentation, we should recognize that our nucleic acids, being one of our major biological molecules, act as our primary source of genetic information when we think about living things. Now, the majority of life contains their genetic information within DNA, or deoxyribonucleic acid, whose primary focus and goal is to store and transmit this genetic information from one generation to the next. So we've already discussed mitosis and meiosis, where we replicate our DNA during the synthesis phase. This to ensure that each of the offspring that we're producing, whether it is a daughter cell or a gamete that can go into making a zygote, that information is maintained in that offspring. Now, we have a slight variation on DNA, which is another type of nucleic acid, which is RNA, or ribonucleic acid. We're going to highlight some of the differences between these two molecules as we progress through this topic. But what's important for RNA is that this is an intermediary in our bodies. So this takes the information that is encoded within our DNA and creates a usable, workable copy that can then go and build us a protein. So this unit is really all going to be about how do we go from that genotype to our phenotype. So this is a great unit to follow up from our previous heredity unit. Throughout this unit, we're going to highlight a couple of these major processes. Look forward to topic 6.3 and 6.4 when we get into both transcription and translation, respectively. So let's talk a little bit more about DNA and its structure. We've previously highlighted the fact that DNA exists within our cells as complexes. And so we get DNA that is wound around these proteins, these chaperone proteins called histones. So histones are like beads where DNA is the string. And together, this DNA histone complex forms what we call a nucleosome. And when we get multiple nucleosomes together, we form chromatin. You may recognize chromatin from some previous topics, but this is essentially our loose organization of DNA as we find it when we're not preparing for division. When we are getting ready to divide, the DNA existing as chromatin will condense, and as it condenses, it will form what we recognize as a chromosome. This is where our DNA becomes visible. So when we look at chromosomes of different species and different types of cells, they have slightly different configurations. Less complex organisms tend to keep their chromosomes as a circular structure. So the end is the beginning. And this is useful for a variety of different things in that it keeps everything all in one place, compact, and we don't really have a whole lot of genes to work with. So in a prokaryotic cell like a bacteria, we may find a single circular chromosome, but we may also find accessory chromosomes as plasmids. Plasmids are like the DLC version of the genes. So these are extra genes that may be acquired from other bacteria. We'll talk a little bit more about that process in the future. And eukaryotic cells can have plasmids too. You get these, you can insert plasmids into a cell and you can make or cause the expression of certain genes that are encoded within that plasmid. Very useful for biotechnology. Now, our chromosomes, and really largely eukaryotic chromosomes in general, are not circular in that we have a lot more genetic information. We have a lot more genes that have been acquired over all of this direction of complexity that we've gone in. So we will have multiple chromosomes. You should know that we have, as humans, 23 chromosomes. 22 are autosomes. One set is our sex chromosomes. And these are largely linear. So we get these rod-shaped chromosomes, which you should recognize from all of the models that we've used to mimic the process of mitosis and meiosis. So let's zoom in to DNA and RNA a little bit closer. When we talk about nucleic acids, we are talking about a polymer. This is a term you should recall from Unit 1. So polymers are these complexes that are made up of repeating subunits, known as monomers, and with nucleic acids, our monomer is the nucleotide. Now, nucleotides themselves are composed of three basic parts. We have a 5-carbon sugar or a pentose sugar that is attached to a phosphate group and to a nitrogenous base. Now, here's where we get our first distinction between DNA and RNA. You'll notice one major difference between our DNA and our RNA sugars, and that is that our DNA has one less oxygen attached to its pentose sugar compared to RNA. Now, the D in deoxyribose means to remove. So we have taken an oxygen from this molecule, and that is what makes this pentose sugar a deoxyribose. Now, you'll notice that I highlighted some of the directionality that is associated with our carbohydrates. This is something that we covered back in Unit 1, but we really haven't used it all too much, so it may be something that we want to refresh our memory with. So when we count the directionality of our sugars, we are going to start with the carbon that is attached most closely to what would be a carboxyl group. So this is our first, our prime sugar here. So this is, this is carbon 1. And then we are going to count in a clockwise direction. So 2, 3, 4, 5. So there's our pentose sugar, penta meaning 5. Now, what's important to identify here is the attachment points that we have. So you can see that our phosphate group is attached to our 5' sugar. And I've highlighted our 3' sugar as well. So this is where we will attach a phosphate group to another nucleotide. So we can create a phosphodiester bond and create a nucleic acid sequence of nucleotides. So one of the main ways that we can encode genetic information, because you'll notice that this part of our DNA molecule, this part of our RNA molecule, those are going to look the same, regardless of what we're dealing with. What will change, however, is this nitrogenous base. So here we have a thymine, and over here we have a uracil. And you won't have to memorize the structures of these nitrogenous bases aside from a few key points. But we can tell our differences between our nucleotides based on the nitrogenous bases that are attached to those pentose sugars. So we actually have two different types of nitrogenous bases. We have the pyrimidines and purines. Our pyrimidines include uracil, which is only found in RNA, thymine, which is only found in DNA, and cytosine, which is found in both DNA and RNA. An easy way to remember which ones are pyrimidines is that we cut the pi. So if you spell pi wrong, it works a little bit better. I also was always one when I was in high school to recognize that two, at least of the pyrimidines, have Y in their name. So if you look at the Y in pyrimidines, then we have the Y in our thymine and cytosine. And then it's a little bit of extra memorization to know that thymine gets swapped out for uracil. You'll notice if you look at all of these pyrimidines that all of these are single ringed in their structure. And that is what differentiates these nitrogenous bases from our purines. Our purines are adenine, and guanine. Both adenine and guanine are found in DNA and RNA. A way to remember your purines, aside from just these ones aren't pyrimidines, is the mnemonic pure as gold. So pure as in purines, as your A for adenine, and gold, your G for guanine. And our major distinction from the pyrimidines is that our purines are double ringed structures. So we get 2 rings in our purines. If we look at DNA and RNA just in general, another big thing that we can differentiate them on, aside from the sugar and the differences in the nitrogenous bases, is that DNA is a double helix. We have 2 strands where we have our nitrogenous bases linked in complementary base pairs in the center by hydrogen bonds, and then our sugar phosphate backbone from either strand. If we compare that to RNA, RNA is single-stranded, so we only have 1/2 of the sequence of information. And this actually benefits all of the processes in so many different ways. RNA can form a whole bunch of different configurations that allow it to play a few different roles in future processes. It also allows RNA to read a template of DNA and build its structure based on that template. So while we may think, oh, RNA can't be as good as DNA because it's only half the information, it is a wildly versatile molecule that you will come to recognize. Now, I left out the majority of the historical research that went into discovering the structure of DNA in this video. I felt that it's not really something that we have to know for the AP exam, but if you are curious, there are a whole bunch of fascinating studies and experiments that were undertaken throughout the 1920s through 1950s in order to figure out what was controlling how we looked and what genes were produced and where all this information was coming from. But one of the experiments that I would like to highlight is that of Erwin Chargaff. So Chargaff essentially, in the 1950s, collected a whole bunch of DNA from a whole bunch of different species, and he wanted to look at the composition of these DNA molecules. So what he did was he used some methods to essentially tag your different bases so that he could count and determine a percentage of the DNA structure that was composed of each of our bases: adenine, thymine, cytosine, and guanine. Now you'll notice that these percentages aren't exactly equal. They're really just more roughly equal, that we have essentially the same percentage of adenine as we do thymine and the same percentage of guanine as we have cytosine. They would be more perfect, but we do have to contend that this was the 1950s and there were some limitations that came along with his methods of calculating this data. But what this tells us is that we can infer that for every adenine, there is a thymine. For every cytosine, there is a guanine. And this allows us to determine, at least looking at data, that we have complementary base pairs. If we have an adenine on one strand of DNA, it is bonded to a thymine. If we have a cytosine on one strand, it is bonded to a guanine. And so if we look at a model of DNA structure, we can see these two strands. We've got our hydrogen bonds in the middle, hydrogen bonds being weak bonds that can be easily broken, but also easily built. This will come into play when we look at DNA replication. All of the rest of the bonds going on in this molecule, those are covalent bonds. We really don't want anything falling apart when it comes to the strands. So the covalent bonds are our strong bonds that can hold everything together. So here are our complementary base pairs. So A with T, the amoeba sisters have the best way to remember it: apples in a tree, and cytosine and guanine, cars in a garage. These will be important to recognize as you may be asked to look at a sequence and determine what the new or complementary sequence would be. It is interesting to note that the two complementary base pair groups do differ in the number of hydrogen bonds that are formed. Between adenine and thymine, we only have 2 hydrogen bonds, and between guanine and cytosine, we have 3. Sometimes it is useful, especially when you've got a limited model, to identify the number of bonds, and that'll essentially tell you which complementary bases we're working with. Like, for example, if I really wanted to simplify my model here, I could swap out all of those nitrogenous bases. We can get a little lazy with our shapes, and so you can have complementary shapes here. But I've still got those 2 hydrogen bonds for adenine and thymine, and my 3 hydrogen bonds for guanine and cytosine. An important thing to recognize is that in order for our DNA to be a consistent width along all of its structure, our strands are actually going to run in opposite directions. They're what we're calling anti-parallel. And so when we look at this anti-parallel structure, and this is where our knowledge of the directionality of our carbohydrates is going to come into play, is that we're running from 3' to 5'. We're running from 3' to 5'. Both of these DNA strands run in the same direction but opposite from each other. They are anti-parallel. One of the ways that we often like to look at DNA molecules, of course, when it's not twisted into a helix, is that we can metaphorically call it a ladder. So with a ladder, you have the rungs in the center and the struts or your bars on the side. And so our sugar phosphate backbone, these are our struts, so these are what are holding everything together. And then our rungs, our nitrogenous base complementary base pairs, these are what are connecting those two sides together. So, with this in mind, in the next video, we're going to look at how exactly we can take this molecule of DNA and create a new identical copy of this DNA with as little mistakes as possible. So I will see you guys back in the next video where we will discuss topic 6.2, DNA replication, and until then, have a great rest of your day.